Hello and welcome to part 2 of chapter 23. Here we're going to start by talking about adaptive radiations. Now this is probably more or less like it sounds. You have a radiation of species as a consequence of the situation where maybe a species or group of species develop a certain adaptation or characteristic, or you have a lot of open available habitat niche, possibly following a mass extinction that creates that open habitat and open niches. And usually what you see is an environment where there are a few other species, so there's less competition for resources, and then also a lot of resources and often varied resources as well. We see speciation events occurring because populations are exploiting different resources, habitats, and so on. So if we think about this on a really big scale, you could think, historically the radiation of mammals after the extinction of dinosaurs. And so that was really important, not great for the dinosaurs, but that created a whole bunch of open niche that mammals could then exploit. And that's then when we see this diversification of mammals in a variety of ways. It isn't that mammals didn't exist before 65 million years ago when the dinosaurs went extinct, or there was a big extinction event on the planet. They were there. Two groups of mammals, specifically marsupials and eutherians. But after 65 million years or so ago, that's where we start to see these branches really expanding and diversification occurring. There are lots of other points in the fossil record that we've really already talked about. We see these important events and then following that, a radiation of species. So just photosynthesis, for example, boom, organisms can photosynthesize now. This is an untapped resource. Changes in proportion of predators in the Cambrian. We looked at that as an example of different species that make it through these, or the changes that can happen after an extinction event. Just plants, insects, colonization of land, etc., etc. Now, in some of these cases, an important event or important factor that we have to consider is called a key innovation. Now, a key innovation is something that allows an organism, a group of organisms, to exploit resources that weren't previously accessible. And a couple of examples would be evolution of lungs in fish, so now exploiting terrestrial environments, and then wings in birds and insects. All of a sudden, they can occupy a completely different ecological niche. And this allows these species to adapt to new parts of the environment. And then we also see, again, speciation events occurring there because they're occupying different environments they previously hadn't occupied, and then there's variation amongst those environments as well. Now, those key adaptations are really important, but also when a key adaptation or colonization or expansion of one species or group of species occurs, that might also mean then another group of species can then radiate because maybe they eat that other species. The example here would be colonization of land by plants, and then this amazing diversification of insects that follow. That, a lot of these insects would feed on plants, and then insects that feed on other insects, and so on. You see adaptive radiations a lot of times going together in different groups of organisms. Just taking plants as an example, you start to, in land plants specifically, you see some key innovations that allow plants to start to do different things, and ultimately being able to grow as form trees. You see diversification of trees, and then the spread of trees all over the planet. A fascinating place where we see a ton of adaptive radiation is somewhere called the Cambrian explosion. And so this occurred about 535 to 525 million years ago. And here is where we see in the fossil record just an amazing diversity of fossils that were not seen before in animal taxa. And that doesn't mean, again, that these things didn't exist before. So sponges, mollusks, cnidarians, those are things like corals and jellyfish. These things did exist, but not to the degree and to the diversity that we see in the Cambrian. It's called an explosion because we just see this massive diversity all of a sudden at this point. Now, I say all of a sudden, but really this is happening very slowly. We're just looking at it as snapshots in the fossil record. As we get more and more fossils and put this picture together, what we see is we see some of this diversification and differentiation of the major phyla, like say sponges and cnidarians, before the Cambrian, which is here on this figure. So these groups of organisms do exist, but again, they're not as diverse at this point. And after in the Cambrian, this is where we start to see this radiation really occurring. Sometimes it's said that the Cambrian explosion had a long fuse because it sort of takes a while for this adaptive radiation to actually happen. All right, so this question's about whether you'd be able to find early Cambrian fossils in New York State. So check out this biointeractive Earth viewer from the Howard Hughes Medical Institute, and I'll provide the link in the question link here, and answer this question. All right, so you would have found, looking at this, 540 million years ago, most of New York State was in the continental shelf of one of the major continents on the planet at the time. So that actually would have been a great place for marine fossils to accumulate. And you can indeed find these Cambrian fossils in parts of New York State. Now, adaptive radiations can occur also at a regional scale, not necessarily just global and through geologic time, but also at a much smaller scale. And islands are often a great example of adaptive radiations, particularly the Hawaiian Islands. If you want to really study adaptive radiation, great place to visit is Hawaii. Not too bad of a place to do research either. So in this example, we see these Drosophila flies. So think, fruit flies, Drosophila, on these various islands in Hawaii. And what you see here are two main lineages of Drosophila. This lineage of 3 species down here on the bottom of the tree, and then these 4 species at the top. And you find these on these different islands here. And this suggests that you had 2 original lineages of flies that then diversified on these 3 different islands here in Hawaii. In this lineage toward the top of the tree here, we actually have 2 different species diverging on the island of Hawaii itself. So, when we're talking about something like a fruit fly, maybe just raw distance or maybe utilizing different resources results in a speciation event, even relatively close to one another. And so there's a few ways that these kinds of adaptive radiations might occur, just for examples purposes. Looking at islands here. So we have an ancestral species, this bird in this case, colonizing this island here. That bird species then could potentially colonize these other islands. And then because they're relatively isolated, you see these different groups of organisms on these different islands speciate. Now, where the adaptive radiation comes in is really this next step. So now, over time, our different island species have become distinct species. They can't interbreed. And now each of these colonizes these other islands. Maybe this yellow-colored bird, these eat a certain kind of seed that these red-colored birds don't eat, and so on. So they're using different resources. We'll talk about that in a second. Another possibility would be that speciation hasn't completely occurred here after our colonization of islands, and then maybe slightly different genetic populations, and then the birds mix again. So these black ones colonize these other 2 islands, the red ones colonize these other 2, and so on. They're slightly different, but different enough that then we see speciation occurring after this sort of recolonization and spread. So these sound very similar, but it's more about when does speciation occur, before we get the mixing of these different populations or species, or after. Either way, you end up with a diverse group of species and an adaptive radiation. So in either example, potentially, character displacement could be very important. And this is all about minimizing competition between resources. Think back to disruptive selection. So that was a key factor with disruptive selection, where we see different organisms exploiting different resources. This is an example showing body size, but think more back to the birds that were eating either large seeds or small seeds and not intermediate sized seeds. That leads to that disruptive selection. And because of that, there's minimal or less competition, and we call that character displacement. So character displacement favors organisms that use a resource that's just not being used, right? That results in greater fitness because there's less competition, and that will be reinforced and lead to speciation. Where we see this as an excellent example are the adaptive radiations of finches in the Galapagos Islands, a very famous example because Darwin studied these finches, and they've been studied at length from that point. And so we have these 4 different general types of finches. They feed on different resources. They live in different habitats. And we see these different lineages. So the ground and cactus finches are a lineage, the tree finches. We've got a vegetarian tree finch. So warbler finches are basal to this group entirely. So a great example of adaptive radiation and character displacement. All right, here's a question for you. Upon being formed, islands like Hawaii should feature what characteristic leading to what phenomenon? All right, yeah, the characteristic would be a bunch of open niche, and then that would lead to adaptive radiation, which is a phenomenon. All right, so let's switch gears a little bit. This could be applicable to adaptive radiation, but is just broadly applicable to macroevolution in general, where we see major changes in body form in evolutionary history. This is obviously linked back to genetic mechanisms. And we know from understanding development a little bit about developmental biology. There are master regulatory genes that tell an organism this is the front, back, head, tail, etc. And the timing of those genes being turned on, or whether they're turned on at all, or whether they're present, or whether there's multiple copies, that's going to determine what sort of the capability of that organism is in terms of its development based on its genetics. So let's first look at this idea of differences in timing of development. So heterochrony, think chronological, that means time. Chrony means time. Hetero means different. And so here's an evolutionary change in a rate of timing of developmental events. So we can use chimpanzees and humans as an example, and we're talking about the genes that are important for regulating skull development in both chimps and humans. So if we look at a human fetus versus a chimpanzee fetus and map that as our baseline, so this square grid, and then we look at what happens to these bones as the skull develops into adulthood. In a chimpanzee, this shape is very different from what we see in the chimpanzee fetus. The human is much less different. You can almost still appreciate that square grid. And so this is all about the timing of certain genes being turned on or off, and the development, particularly in these jaw bones, in adult humans versus chimpanzees. Another really cool example are the finger bones in bats, which are obviously very long, and they have to support these wings. And so the timing of those genes that are responsible for development of the finger bones, those are going to be turned on for much longer, and that's going to result in increased growth of those finger bones, and you end up with something that looks more like a bat. Another type of heterochrony is called paedomorphosis. And what we see in paedomorphosis is that the organism matures or reaches sexual maturity, or the development of reproductive structures is much quicker than somatic development, which is all the rest of the tissues or body. And what that means is the sexually mature organism retains body features that appear juvenile-like. Now, for that particular species, that's how it looks here, like this axolotl. And so we would connect it back to an ancestral species. We'd say, well, in the ancestral species, these were not juvenile, or these were juvenile traits. But in, say, this axolotl, these are not juvenile traits. This is what this looks like as an adult, but we consider this paedomorphosis because these are what we would consider juvenile traits. So let's look at some of these genes that are important for development. We talked in a previous chapter about homeotic genes. These are those master regulatory genes. Different sets of these are switched on in different regions of the body, which determine in an insect whether, say, legs form in this segment or they don't, or where birds' wings develop, etc. And so these are really critical for determining where certain structures develop. Hox genes in particular are critical for positional information, that where, a certain, say, appendage might develop. And we know this because if you force the expression of Hox genes in a different place, we'll see that body parts can be produced in that wrong location. And this would be the same in plants. So plants don't have Hox genes per se, but they have these MADS box genes that have a similar positional effect in terms of where flowers form on plants. All right, so if an organism has a relatively large number of Hox genes in its genome, which of the following is true? Okay, yeah, so if the more Hox genes that are present in a genome, typically that tends to be associated with much more, relatively more complex anatomy in an organism that might have more Hox genes versus one that does not. Now, these interactions are involve also not just turning things on, but switching things off. So we see diversification of Hox genes as well in different organisms. And here's an example here where we have this UBX gene, which we can just call, Hox gene 7. And in this crustacean ancestor, this is important for all these body segments where UBX is expressed. That makes legs, basically. But in present-day insects, where you have UBX being expressed in, say, these segments here, like in the abdomen, that's turning off leg development. And so these have a diversity of function. And again, potentially the more there are, whether it's saying turn this on or turn this off, again, the more complex body form that we might see in a certain species. Now, these regulatory controls might be occurring at a very small scale, not just like across the whole body, but in very specific regions. And the example here in your textbook is of three-spine sticklebacks, marine versus freshwater, and the expression of this PITX1 gene. So where we see expression, you have this blue color, and in the marine sticklebacks that form these ventral spines here, you see expression of those genes just in this very spot, and that's going to allow the spine formation. Whereas in these freshwater sticklebacks, you don't see any expression at all, and these fish don't form those spines at all. All right, so here's a question. All right, yeah, so if these flies are expressing the UBX gene that suppresses appendage production, you're going to end up with a mature organism that, say, doesn't have legs. It's going to resemble a maggot. That is an example of pedomorphosis. So let's look at an example of an evolutionary novelty, the evolution of an eye, which is just fascinating. This is sometimes cited as an example of how could an eye have evolved? It's so complex and you have all these parts. It seems like just by chance and natural selection that how could that have happened? Well, it could have happened in a very stepwise way. And we actually see in extant organisms that are still living today, different eye types that are representative of probably what eyes looked like in past organisms in the fossil record. And so we can start with this patch of photosensitive cells. So this is the most simple eye. You find these in organisms like limpets here. And this kind of eye is really basically good for distinguishing light and dark, but not really forming much of an image. In a type of mollusk here, we see this eye cup type of eye where you have these photosensitive cells kind of forming this cup-like structure. Further along here in nautiluses, you find a fluid-filled cavity and a space here that allows only light passing through here, just like an iris. In another type of snail here, this marine snail Murex, they have a similar eye to that previous type of eye, but the difference here is you have this mass, which is a lens. So you're actually having this organism being able to focus the light that's coming into that eye and meeting these photoreceptive type cells. Another kind of eye that we see in another group of mollusks, the squid and octopuses, this is much more complex looking. We have those photoreceptive cells, the fluid-filled cavity. This lens is very distinct. It can help to focus and collect light. And this is fascinating because this is actually very similar to a kind of eye that we see in mammals. And this has evolved independently. So you can imagine then thinking back, like starting with an organism that has sort of those basic photoreceptive type cells, you have the building step by step of these structures toward a much more complex eye in different lineages. And now that, again, doesn't mean that eyes are inevitably evolving toward a certain type. It's just that there's the capacity that this can occur given the right set of circumstances. For, say, the limpets, for example, those are extant organisms. They're around today. They just have that photosensitive patch of cells. That's sufficient. It doesn't mean that limpets are eventually going to have a much more complex eye. It's just that it can happen in some lineages. Now, another interesting feature are acceptations. And this sounds very simple, and we'll use the example of feathers here. So birds, or dinosaurs, really, evolved feathers from scales, probably as a mechanism for thermoregulation, to keep warm. But this eventually allowed for flight in birds. And so the original function, if you will, of feathers was just thermoregulation to keep warm, but then evolved into flight. This doesn't mean that there was feathers were eventually going to be important for flight. It's just that feature, feathers, was useful when flight arose in birds. And it's really important to remember, natural selection can only work on existing variation. And so the fact that those were structures that could be quote unquote built upon by natural selection was certainly beneficial. But sometimes this does get confused, again, thinking that, this is something that was sort of meant to happen so that you end up with flying dinosaurs. No, it's just again, coincidental that that happened, and that existing trait, which was just for thermoregulation, was important for this other adaptation. All right, let's see if you can figure one out here. Which of the following is an example of an exaptation? All right, hopefully a pretty easy one. And let's build on this same idea that where exaptation sometimes gets confused, that we sort of quote unquote working towards something. Absolutely not. Evolution is not goal oriented. Evolution just tinkers with existing variation. Maybe new adaptations occur, and that's a real benefit, and there's adaptive radiations, but they're only built upon what's already there. And you've already heard me say this in earlier lectures, but this is just a reminder. There's no drive toward a certain phenotype, that we don't see evolution giving creatures what they need, like this polar bear is naked and it needs a fur coat, right? It's just this interaction between organisms and their environment, differential reproduction, reproductive success, and so forth. And so it's important as, biologists, where, evolution is just fundamental to understanding biology. We're, a biologist, we're all evolutionary biologists to some extent. But it's important to take in account total evidence. And a good example of this is from in the fossil record, where we have patchy information in some cases, more complete information in other cases. And we need to really look at as much information as possible. And the idea that if you sort of cherry pick data, which would be bad science to begin with, you might get led to the wrong conclusion. And the example your textbook gives is on horses and development of these single-toed organisms. And if we look at the evolution of horses in the fossil record, and say we look at these 4 horses here on the right, we see this trend toward having fewer digits, and it looks very much like a linear trend. But we're excluding, say, these other lineages over here where we don't see the reduction of that forelimb to only having 1 toe. And so there isn't just some linear pattern here that we're following. We need to take into account all the branches of this phylogenetic tree for understanding horse evolution. And there would have been several places or several branches here that maybe, have gone extinct where we didn't see that trend toward a single toe in these animals. But in modern horses today, that is what we see. But there were these other branches where we didn't see that occurring. And here's the figure from your text showing a little more closely the limbs of these animals, where we see different numbers of digits. Again, in modern horses, we just see the 1 toe, but we do see multiple toes in these other lineages. And so this brings up this idea. It's important when we're thinking about the tree of life, everything extant living today are out on the tips of these branches here. And if we're trying to understand the evolution of how to get to one of these tips, say we're interested in human evolution, yes, we might be interested in following this track back here, way back to the beginning, the root of the tree. But look at all these side branches. We need to consider those as that total evidence, including these branches that go kaput. They don't reach that outside of the tree where all the living things are today. All right, so we'll wrap up this part with this idea of something called the species selection model. So species selection is really all about considering extinction and speciation rates. So extinction's always occurring, speciation's occurring. It's which one is winning out. So looking at, if extinction is exceeding speciation, that lineage is that group of species is heading downhill, and vice versa, we start to see increasing numbers of groups of species. You can think of it a little bit like relative fitness in a population, that we covered that when we're thinking about within species. And so here we're taking that up to the level of species where we see differential success of different groups of species over time. And this is a much more important at that macroevolutionary level. The example here, we've got these 3 different types of snails. The ones that are shown in blue and red here have a really big range. The reason for that is their larval dispersal. So those are just cast out into the ocean here, and so they can spread very wide. So having a really broad distribution is sort of banking against an extinction event, right? So if organisms go extinct around Papua New Guinea, that's okay. There's still lots of other populations all throughout the range here. But a downside of this, maybe less risk of extinction, sure, but because dispersal is so good, there's also the chance of lower speciation because there's so much gene flow because these things can move around all over the place. Let's contrast it here with this other snail species shown in black. This one does not have a dispersive life stage at all. And so we have these 3 distinct populations, genetic populations. So here, the odds of speciation are high because you don't have gene flow necessarily between these populations. But the risk of extinction, because this much smaller population, say these islands over here, the risk of extinction in that particular place might be higher. And so it's kind of this trade-off of speciation and extinction, and the net gain or loss is really what that species selection model is all about. All right, that brings us to the end of Chapter 23 and to the end of the content for this semester. Good luck finishing this up and finishing up with the rest of this course.